Hi all, hi everyone, and welcome for participating in this new AITF alumni web series. I'm Lucas Perez. I'm a strategic expert and author in health and aging solutions, and I'm also the French representative for the AITF alumni network. It's a pleasure to have you today in order to address with Roberta Menezes, Chief Marketing Officer at Tech, what is this amazing and mysterious science called microfluidic. We're going to present you why and how microfluidic could revolutionize the field of healthcare in terms of research and in terms of development. Then you will have the opportunity to ask us some questions during a Q&A session. And for that reason, please to put your question in the chat before and during the session. So what is the AITF alumni and what do we offer to our alumni? The AITF alumni is an amazing community gathering more than 2,000 learners, academics, and entrepreneurs all across Europe and beyond. And in joining the network, you will have the opportunity to learn more about professional skills, teach and share your experiences and expertise, co-create a business through an entrepreneurship program, for instance, build your network, and also get a job. For that, you have to join our alumni platform, where you can see our members and our network all across Europe. And you can also meet the AITF alumni board, where each of us represent a country or a group of countries called a region. And from my side, I represent the French alumni team, and with my colleague and friends, Lilian, Ben, and Soufiane, we build some events and we try to share of ideas in terms of healthcare and autonomy. So don't hesitate to join the AITF alumni network now in scanning this QR code, and let's together the future of Europe. Before to pass my time to Roberta, I just want to share you a quick insight about microfluidics and aging. As you know, the senior population is continuously aging in the world, and demand for novel strategies to repair and regenerate damaged and diseased tissues and organs has grown. The desire to repair and regenerate the body, the whole body, is more and more possible thanks to the transplantation of patient-owned tissue, for instance. However, limited availability becomes a problematic issue, especially for this senior population. As a consequence, the field of regenerative medicine has emerged to address this growing demand. But what is regenerative medicine? Regenerative medicine is a global and interdisciplinary field of research and clinical applications focused on the repair, replacement, and regeneration of cells, tissues, and organs to restore imperfection resulting from aging, but also from a trauma and disease. This is where we can use a sort of lab or organ on chip in order to improve this field of regenerative medicine. And here are two examples in terms of research on regenerative processes, two examples of in vitro and not in vivo research, because it works on chip. The first is musculoskeletal regeneration. To give you an example, today in Holland, a group of researchers in physiology are working on skeletal muscle on a sheep in order to reproduce a muscle disease in vitro out of the body, out of the human, and at the end to develop a new therapy and to test the patient's specific response to a therapy, but on a chip. And the other one, example of microfluidic systems and the microfabrication is the research in terms of neuronal regeneration. And for this field, researchers are also working on neuronal regeneration, and they try to manipulate neurons in a very, very small environment, and which is very exciting to understand and prevent degeneration after, for example, a head trauma caused by an accident. So thank you for listening, and I let Roberta present us in detail what is microfluidic and what Eden technology offers in this field. Thank you, and I think it's your turn, Roberta.
Thank you so much, Lucas. So I'm gonna set up my presentation share. Let's see here. Well, thank you so much for that introductory information on microfluidics and some examples for aging. I'd just like to start off by thanking you for inviting me to do this talk, and thank you for all of the people that are attending. Hopefully this will be instructive and interesting for you. As Lucas mentioned, my name is Roberta, and I'm Chief Marketing Sales Officer at Eden Tech. And we are a microfluidics company focusing on user-friendly microfabrication solutions to help boost device development and the commercialization into industry. So today I'm hoping to give you a little bit of a taster of what is microfluidics, what are the advantages that it can bring for innovation, but more particularly in innovation for healthcare and medicine. So here I just have a layout of the presentation. I'm going to start off by explaining microfluidics, its advantages, and the different applications in healthcare. Then I'll go into different methods of cell culture that are really important to ensure the correct maturity and cell morphology of the cells that are in the device, so that we can accurately, or at least try to more accurately, imitate the physiological microenvironment. Then I'll cover some specific examples of microfluidic devices for different healthcare applications. And I'll move into what are the microfabrication challenges, the material challenges that will help us move microfluidic technologies more easily into industry and into commercial use. And finally, how Eden Tech wants to help overcome these challenges with our microfabrication solutions. So what is microfluidics? Simply put, it's the manipulation of fluids in micrometric spaces. And this can be done in a variety of different materials, from paper to gels to different types of polymers and plastics. And microfluidics has many advantages. First, there is the handling of a small volume of reagents. So this could range from the microliter all the way down to the femtoliter. And by handling small samples, we're saving precious, sometimes costly samples, and we're also allowing for the parallelization of reactions, which can be useful in screening or in the studies of single molecules or cells. Next, we're dealing with a closed system, which then diminishes the risk of contamination for the experiment, but also improves the user safety. Because we're handling such small volumes of liquids in a micrometric space, there is also the advantage of having high volume to surface ratio, and this shortens the reaction time, but it also allows for a laminar regime, which means that there's no mixing of fluids. And this allows for better control of the kinetics of the fluid and of the particles or molecules that are inside of it. And that way we can better control for the location and the transfer of these molecules, and for example, create concentration gradients. And finally, the fact that these are handheld devices means that they open up the opportunity for point-of-care testing without a laboratory infrastructure. Now, the majority of microfluidic applications in the market thus far are mainly concentrated in the medical field, and these can be categorized firstly into clinical diagnostics, which are devices that are handled by expert trained individuals and with laboratory infrastructure and heavy equipment at times. In contrast, we also have point of care diagnostics, which are devices that are made for the end consumer use. So this is a person that hasn't had specialized training. And these are ideally also used either with a small platform for imaging or analysis, or just in a colorimetric way so that there is no additional equipment needed. Next, we have drug delivery devices, which are essentially in microneedles or other devices that deliver therapeutic agents. And finally, we have devices that are tools for pharma. These include cell analysis devices, devices for tissue engineering, drug screening, drug development, and these are often devices that handle cells and cell culture, and that can have quite a complex design. So since there is such a wide array of applications for microfluidics, today I've chosen to focus my talk on the devices for tools for pharma, because they're particularly interesting in the sophisticated cell culture techniques and the complex device designs. Now, the first thing that we must mention is that our cells provide us a wealth of information on our health, development, and pathologies. So the cells can provide us information from the genomic or DNA level all the way through to the phenotypic or observable level. And the types of cells that can be used for these types of studies can be commercial preserved cell lines, or they could be, for example, extracted stem cells, which then have to be cultured and differentiated into other types of cell types. Now, when we have a look at classic 2D cell culture methods, these cells are cultured on a rigid flat surface, which creates a homogeneous monolayer of cells. Now, this is a very high throughput process that is composed of standardized protocols, is rapid, allows for easy cell analysis, is low cost, but at the same time, it's time consuming because it requires manual exchange of medium, which supplies the nutrients to the cells, and also the removal of that waste that the cells are generating. But also the environment in which the cells are being cultured is simply not complex enough to really imitate or mimic the physiological microenvironment in our bodies. And this is because it lacks 3D structures, mechanical forces, or signaling and interactions that occur within our bodies. So to further boost the relevance, the physiological relevance of cells which are cultured, researchers came up with static 3D cell cultures. And here, essentially, we are culturing the cells either suspended in a liquid environment, such as the hanging drop method that you can see illustrated here, or with the help of a scaffold, which could be a gel matrix, for example, in order to allow the cells to assemble in a more complex cytoarchitecture. And these types of methods are high throughput as well, and they allow for a more complex mass of cells to be formed and to be more representative of the physiological microenvironment. However, there is no regeneration here. So there is a formation of vascularization, but in most cases, these vessels are not functional. So there is no regeneration, and there is no internal perfusion. What does that mean? The nutrients that are supplied to the cells will be able to nourish the cells at the surface of these masses, but not internally. And so although we are getting to a higher physiological relevance, it is still quite far from mimicking the physiological microenvironment of our bodies. So in order to further optimize these cell culture processes, microfluidics has been used as a tool to reduce manual operation, provide various functions within one device, but also to provide real-time monitoring. So this could be using fluorescence or sensor integration. And so for the first time, what we can do is actually visualize biological processes within a device. It also provides a way to grow complex ensembles of different cell types and incorporate physical aspects that allow us to imitate the physiological microenvironment. By that, I mean is that there is there are 3D features that can be incorporated into these devices. So for example, scaffolds, membranes, microfeatures within the devices that allows us to mimic the mechanical properties of tissues. However, at the same time, these devices have a high cost and they're usually quite complex, meaning that it's difficult to produce them in a high throughput manner. So now what I would like to do is cover specific examples of these microfluidic devices for cell culture, which range from single cell analysis devices all the way through to organ on chips. Now, single cell analysis devices are interesting for applications like the study of microbiomes, in which there is high population heterogeneity, all the way through to organ on chips, which are devices incorporating different cell types in a particular spatial arrangement in order to mimic the physiological microenvironment. And so with that, I'm going to start off with an example that's linked to what Lucas talked to us about, which is aging studies. And what I thought was particularly interesting about this device that the authors devised is that they really tried to use microfluidics as a tool for automation of classic laboratory protocols. So here we have a device to study the worm C. elegans, which is a model organism for aging studies. And it is a worm that's about 1 millimeter in length and 80 microns in width that has been found to have a single gene mutation that allows for lifespan expansion. And the worm C. elegans is particularly interesting to study because it has a relatively short lifespan of 20 days. It is simple to prepare, let's say, genetically similar populations within a 1 to 3 day protocol. They are transparent, allowing for fluorescence microscopy studies. But the classical experimental protocols for C. elegans are quite complicated in that the worms need to be isolated and dissected manually, and a variety of different chemicals are used to fix or immobilize the worm. So for example, adhesives or anesthetics, and other chemicals, and these compromise the natural lifespan of the worm. So what the authors did here is devise a relatively simple device which can automate the process of synchronizing parent populations of worms and then studying individual worms through high throughput, high resolution fluorescence microscopy. So if we have a look at the device, you can see that in the red section, there is a chamber, and this chamber is surrounded by filters that serve to filter out the progeny of the parent population and collect them separately, keeping the generation synchronized. Then we have a section highlighted in the blue dotted line, which allows for the trapping of individual worms and their study via fluorescence microscopy. And how the worms are transferred from one section of the device to the other is thanks to the microfluidic lines, which flow the liquid in opposing directions. And with this design, they were able to not only study the cellular and subcellular characteristics of the worms throughout their entire lifespan, but they were also able to keep the parent population synchronized, and they did it all within a drug-free environment. And this is a really clear example of how microfluidics can make experimental protocols more user-friendly through automation. And a device like this can then be used to study other types of cells, and several devices can be used to parallelize the process. So next we have an example of microfluidics for drug development. And here I chose the liver chip device that's commercially available from Emulate. And this is a great example of how organ-on-chip devices or technologies can be commercialized to further help the development of research to understand organ function, but also to screen for drugs and test how these models actually compare to classical animal models. Now, we know that the drug development process is quite a slow, costly, and strict process that has seen the gradual number of approved drugs over the year continuously decrease. And here what the authors decided to do is to use this model for a liver to study drug-induced liver injury. The way they built the device was to mimic the layered structure of the liver, which is composed of hepatocyte cells intercalated with other different cell types. So in the simplistic model of the chip that you can see below, there are two channels that run through the device. In the upper channel, we see the hepatocyte cells, which are embedded in an extracellular matrix, which provides this structure surrounding the cells that will more accurately mimic the microphysiological environment. And this channel is then separated from the bottom channel by a porous membrane. And in this bottom channel, we have other specialized cells, such as endothelial cells, that are important for aligning the vasculature and capillary structures. We have Kupffer cells that are important in protecting the liver against infections, and we have stellate cells that control for blood flow. So in this simplistic model, what they did is first study the combination of the different cell types and how they relate to organ function. They did a 2-cell model using a combination of hepatocytes and endothelial cells, and they did a 4-cell model, which had all of the cells cultured within the device. And with this, they were actually able to study the cellular signaling of the different cell type combinations to make sure that the device actually functioned and expressed the correct signaling molecules that are usually expressed for these types of cells in the organ. And they were able to study the drug metabolizing capabilities of these cells over extended periods of time, and found very interesting results that confirmed that the cells within the device can actually metabolize clinically relevant concentrations of drugs in the same way it has been observed in humans in clinical trials, but at the same time, either confirming or negating what classical animal models have produced. And so showing that these devices can give more insight into what are the reactions to drugs that we can observe. So another interesting aspect is that these devices provides continuous microscopy imaging of the entire process. So the observation of the biological processes in the device, but also the observation of the drug-induced liver injury process in the device. And they did this by studying different types of drugs and even drug candidates that had not that were blocked during the clinical trial process, for example, from Janssen Pharmaceuticals. So I chose this example because it is a really interesting case of the standardization and commercialization of an organ-on-chip device that can then be provided to end users for different applications and to study different organs. Now, for the third example, we have a slightly more complex design that is not commercialized and is not available to the end user in a standardized way, but it is also an organ-on-chip device. And here what we're doing, or what the authors wanted to do, was to replicate the tumor microenvironment during cancer development. And these types of devices are particularly interesting to study poor prognosis in the disease development and the sensitivity of a tumor to therapeutics. Now, this device has a seemingly highly complex design. Well, actually, I would say that it is complex. It's composed of a middle chamber that you can see labeled below as the vasculature chamber. And in this chamber, we're going to load certain cell types in order to create a microvasculature, which is really important for the development of a nascent tumor. Why is that? It's because the capillary or this vasculature is going to be responsible for supplying the tumor with the nutrients it requires to grow. Now, it is known that in the arterial end of the capillary, there are diffusion forces that force the nutrients to flow towards the tumor. At the same time, the tumor, in order to be able to nourish itself and signal to the capillary to release these nutrients, it needs to release signaling molecules called morphogens towards the capillary. Now, this means that this tumor signaling molecules have to travel against a gradient. And so what the authors wanted to do here is replicate this complexity of mechanical forces that occur in the tumor microenvironment, as well as the distances. And so here, what they do is they first load the microvasculature into the central channel. And then in the two side channels, what we have are tissue chambers, for which once the microvasculature has developed to a sufficient amount, we are going to load different types of cells that we would like to test. So these are either different types of tumor cell lines, which they tested, or patient-derived tumor organoids, which are these self-assembling cell masses that have been actually extracted from donors and grown to represent the tumor microenvironment in an individual. So here what we have is a device actually that is capable of not only studying the interactions between the tumor cells and the tumor organoids, but also the interactions of those and the microvasculature, which is extremely important for that development. And in this study, what they did was that they monitored the communication between these two, and they also studied their reaction to different types of drugs, including chemotherapy agents and anti-angiogenesis drugs that will impede the development of tumors in typical cancer treatment. Another interesting aspect of this device is the amount of, let's say, high resolution imaging that they could obtain throughout the entire process. So they were able to transduce the different cell lines and cell types with plasmids containing fluorescence in order to be able to visualize the entire process throughout. And they also then did immunofluorescent staining in order to observe particular endpoints of different stages of the tumor development. So this is a much more complex device that really incorporated the interactions between different components of the tumor microenvironment, their development, and their reactions to the introduction of drugs and other stimulus. So now that we have covered different examples of microfluidic devices for tools for pharma, what are the key considerations that we must consider when building such a device? Well, these can be simply categorized into biological requirements, but also technical and fabrication requirements. So within the biological requirements, first you have to really think about what are the different cells that you're trying to study. Are these individual cells? Is this a cell population, or the interaction between different cell types? Also, what are the culture conditions for these cells? Not only in terms of the medium and the nutrients that are supplied, but also in terms of the mechanical forces that should be included. For example, shear stress, compression, stretching, also in terms of the physical chemical factors, so in terms of pH, temperature, and the interactions that occur with cell to cells or cell to molecules. These all have to be considered into the conditions that will allow the cells to best mature into the correct morphology. But also, how will the cells be integrated into the device? That it means the loading of the cells, and for this you need microfluidic lines and inlets and outlets to seed these cells, but also what will be the 3D arrangement? Will we be working with these organoids that I mentioned that are self-assembled cell masses, complex cell masses? Or are we working with a 3D arrangement of channels separated by porous membranes so that we can more closely mimic the mechanical properties of the tissue? These have to be considered in order to have the most, an environment that most closely mimics the physiological microenvironment. But the ability of the cells to mature correctly is also closely tied to the materials used for the fabrication. So for example, there are some basic material requirements such as biocompatibility to ensure that there's no toxicity towards the cells. For example, softness of the material to better emulate physiological environment, and differing levels of gas permeability depending on the cell type and the cell culture time period. Then it's also important to consider how you will analyze the biological processes. So here we saw examples of fluorescent imaging, and for this, the material needs to be optically transparent. That's very important. But then it's also possible to integrate biosensors. And for this, there are materials that have a more standardized and well-known procedure for biosensor integration. So all of that needs to be considered in the material that is selected. And finally, what is the end goal for the production rate? Is this a device that will be continuously produced at small scales or medium scales, or are we going to mass produce it for commercial purpose? In that case, it needs to be a material that serves and that is compatible with processes of production from all scale ranges. And now I want to cover more specifically what are the different fabrication challenges and the material challenges that are associated with the development of microfluidic devices for commercialization. Now, it's really important to make a parallel here between microelectronics and microfluidics. These are both fields that provide efficiency in volumetric integration, parallelization in high throughput, and miniaturization. And microfluidics actually found its origin in microelectronics. The first material used for microfluidics was silicon, which was readily available and was found to have a thermal and chemical stability. But then in the 90s, what researchers did was turn to polymers, which are lower cost, biocompatible, and transparent, which made it really easy or much easier to develop microfluidic applications. Now, beyond the materials, microfluidics is also similar to microelectronics in the evolution of the field. So when microelectronics first developed, there were only, we could only imagine the array of applications that silicon integrated circuits would have in all of the electronics that we use today. And integrated circuits would only have maybe a few thousand transistors incorporated. However, this field has seen an exponential miniaturization, and now there are up to several billions of transistors found in one integrated circuits, with circuits measuring as small as a few nanometers. However, there is a challenge in the material here, in that the silicon atom measures 0.2 nanometers, and this means that it will be difficult to shrink past this scale for the technology. And so now researchers are considering other materials for the development of integrated circuits. And microfluidics, on the other hand, is also seeing some material challenges, as the complexity and the array of different applications for microfluidics continues to increase more and more. Right now, most of the applications are medical related, but there are many other industries that can benefit from microfluidics, such as environmental sensing, optics, and so on. And microfluidics is also seeing a challenge that is related to the materials that are currently used because of this. And so now I'm going to cover more specifically the polymer microfabrication process for polymer microfluidic devices, to explain a little bit what are the material challenges for microfluidics. So if we have a look at this process, we start with the device design, then the fabrication of the master mold. Then we move into what is the device replication, in which the master mold is used to replicate the device. And then the device is assembled with a substrate and many other components, such as electrodes, maybe reagent storage blisters. And after this assembly, if we want to commercialize the device, all of these components have to be compatible with mass production processes. And if we look at the materials that are currently used in the microfabrication of polymer microfluidic devices, we can see firstly that for master molds, SU8 and silicon wafers tend to be one of the preferred methods, especially at small scale and prototyping. However, these wafers are fragile, expensive, and they're certainly not compatible with large scale production. Then when we come to device replication and assembly, there are two general families of materials that are preferred, PDMS in the academic circles and thermoplastics for industrials. Now, PDMS is really attractive for biomedical applications because it's a biocompatible material. It has soft elastomeric properties, it's transparent, and it's accompanied by a relatively straightforward protocol that is easy to learn, especially for biological researchers. However, it still remains relatively expensive. It has low precision in the replication fidelity. It has certain material properties, such as the absorption of hydrophobic molecules, which can affect the experiment and its results. And it is challenging to scale up with because device assembly is usually done in a manual way. And so that is why industrials tend to flock to thermoplastics, because they're compatible with these large-scale production processes, but at the same time, they have challenging mold fabrication because they utilize metallic molds that are very expensive, and it makes it difficult to optimize designs and have different design iterations for testing. And then at the same time, they implement harsh chemical or temperature bonding treatments, which can compromise the device features during the bonding process. And it could also affect any functionalization of surfaces and the interior of the device, which is usually the case for biomedical applications. So currently what we see is kind of a family of material or a particular material, PDMS, which is very popular for this initial steps of prototyping and small scale production, and which was the material used in all the examples that I mentioned before, but that is difficult to scale up with. And then on the other hand, in industry, where we have the use of a material that is difficult to prototype with. And so there is a lack of one material that can really take the researcher from beginning to end of the device development process. And so this is what we're trying to do at Eden Tech, is really reinvent microfluidics, make it more user-friendly, and provide microfabrication solutions that help a smoother, more seamless development of microfluidic devices from small to large scale production. And we do this through by providing biocompatible and scalable materials specifically designed for microfluidics, but also user-friendly equipment, especially for the prototyping small scale production phases before moving into a mass manufacturing. And just to highlight our key materials, we have FlexTime, which is really the first material that has been designed and created specifically for microfluidics and biotech applications. And we have our Sublime machine, which is a portable user-friendly machine that allows initial small to medium scale production of microfluidic devices. And how do these work? So the FlexTime is a thermoplastic elastomer that is certified biocompatible, that has low absorption of hydrophobic molecules. It has soft elastomeric properties, just like PDMS, optical transparency, and it is on top of that recyclable. So the FlexTime sheets, they can be used, molded, sterilized, and then reused again. So that was a really important aspect for us. And then it also has the advantages of thermoplastics. So FlexTime gives an array of production options. It can be fabricated using hot embossing method, 3D printing, row-to-row embossing, and injection molding, meaning that once you start prototyping with this material, you're sure to be able to move on to larger scales of production. And finally, as I mentioned, we have a hot embossing machine, which is providing a user-friendly way of prototyping and very fast and efficient protocols for the researcher to be able to spend less time in the lab and more time thinking strategically about their product development. And the Sublime more concretely provides a 3-minute or less replication protocol, which is really just the application of pressure and temperature on the FlexTime material to replicate the device. A very fast as well assembly and bonding of the FlexTime devices in a 1 to 3 minute step as well. This is also a treatment-free bonding and assembly process that does not implement any harsh chemicals or temperature. And it is priced in reference to other materials so that it provides a competitive advantage, but is also affordable at the same time. This is really important for us to make sure that polymer microfabrication can be a straightforward, user-friendly and fast process for the user. So with that said, I would just like to thank you all for listening, and I'll be happy to exchange with Lucas now for a little bit or accept any questions.
So thank you very much, Roberta, for your presentation. It was really interesting to understand and to know more about the history of microfluidics, this brilliant analogy between microfluidics and microelectronics. And it's really crazy to see that we can create, we can replicate any type of organs, like a liver, heart, on a chip, even a tumor. It's amazing. And you also highlighted the fact that there are many stakes for research and the industry, the laboratory research and the industry. And the key point will be, of course, the mass production. And on this line, I got a question, my first question for you. You didn't tell us about the price of the device or globally of your solution, because there is maybe a software part or some services around the device. And my first question is, what is the cost of your device or globally of your solutions?
So thank you so much for that question, Lucas. I think it's actually quite important to clarify that our solutions are really tools that we're providing researchers to be able to develop their microfluidic technologies, whether that is in the form of the material. So we're really taking into account the properties of the material that are required for biotech applications, and the microfabrication equipment, and making it user-friendly, easy to use, and which boosts the high throughput and efficiency for the user. Now, so unlike a company like Emulate that will provide a ready-made chip platform in which then the user can see the cells of their choice and do the study of their choice, we are providing really the raw material tools for innovators to be able to have full control over this device production process from the beginning to the end. So the material in itself, we really wanted to make it give researchers that competitive advantage, not only in terms of cost, but in terms of time and convenience or peace of mind, really. And so if we take the material, the flex time in raw material costs, we're really looking at a material that is going to be half to a quarter of the price, for example, of PDMS, with the advantage that you can scale up. And that is going to be very similarly priced to other thermoplastics like polycarbonate, COC, polystyrene. So we're really combining the best of those two products, taking advantages of PDMS and the processing advantages of thermoplastics. So the raw material is priced in this way. Now, if you want to get started with the FlexTime, what you can do is just get a full package of the material plus the equipment. And here it's really a minimal investment. So we're talking about in the order of a few thousand, and you have everything that you need to get started and start prototyping your devices and test out your material. And even if you're not ready to invest, for example, in the Sublime hot embossing machine that we offer that I mentioned, it is actually possible to test how quick and easy FlexTime replication is simply by applying a weight on top of the material and inserting it in an oven. And here you will see how easy it is to replicate your design for your device. Then when we're looking, for example, at the smallest amount of FlexTime that you can buy, which is in the form of our starter pack, the cost for a credit card, let's say size sheet, is going to be very comparable to the cost of PDMS for a typical device of the same size. So we're talking at maybe in the order of $2 or $3, which is comparable to PDMS. But what you're saving is the time and the convenience, because it, like I said, it's a 3-minute replication protocol. It's a 3-minute assembly protocol. And on top of that, you don't need any additional equipment for the bonding. There is no treatments, there is no harsh chemicals, no harsh temperatures needed, which makes it ideal for these biotech applications.
Okay, thank you. So the point is more about, yes, the time saving than the price, and it was clear. So we got a question from the audience, and it's Corentin. And Corentin said, can FlexDim prototyping also be performed by people unskilled in microfluidics?
So this is really what we're trying to do at Eden Tech. We're trying to show that microfluidics can be a more user-friendly and a more easy to learn process, which is why we have developed, actually our CEO, Emmanuel Roy, he developed the FlexDim material at the CNRC in Canada, and then founded Eden Tech on this technology. And it's really based on the principle that it's a material developed specifically for biotech applications, which we all know is a really interdisciplinary, microfluidics is a really interdisciplinary field where often biologists, I can say, at least for myself, I came from more of a biological background, and we have to learn these microfluidic skills. And so by making the protocol and the production and fabrication process simpler, we leave the researcher to spend more time focusing on what are the other more, let's say, intellectual skills that are required for fabrication. So the design of the device, simulating, modeling the mechanical forces that are inside of the device, and then democratizing the field in a way.
Thank you. Thank you. So on this line, I got another question, maybe more focused on the regulation and the law related to these microfluidics systems and the microfluidic field. So my question is, are there some law or regulation issues related to microfluidic, such as bioethics, and in which countries or in which region, for example, in Europe or in US?
Okay, no, I think that's a really important question, because I mean, we are talking about devices in which are serving potential applications in, for example, the studying of donor cell samples, or for precision medicine, in which we are, using an individual's cells. And what is important to know to start off is that there has been an enormous amount of investment in this field during the last decade. So, for example, in the US, there has been huge investments coming from DARPA, the NIH, the FDA, to better develop organ-on-chip devices and use them as tools for the pharmaceutical industry. And thanks to these investments, there have been organ-on-chip models that even mimic or try to mimic the a human on a chip, and incorporate up to 8 or 10 organ-on-chips within one entire system. And they're investing heavily in this. In Europe, the Horizon 2020 program created the ORCID strategy, which is a strategy for the development of organ-on-chip technologies, and it incorporates several building blocks, which are the qualification of these devices, their specification, their validation, but also other general topics like ethics and the communication of these types of technologies. And within the ethics segment in particular, the issues that are highlighted are really, as I said, the use of donor-derived cell lines and biopsy tissue, but also the dilemma between standardizing such a technology, but also making it a profitable technology at the same time. So meaning that if we want to standardize it to serve a mutual benefit for all of the pharmaceutical industry and to help progress society and the study of disease. But at the same time, we also want to make these profitable. And so there is a huge ethical dilemma in that, for example, if there is one company that manages to really standardize and mass produce these devices, how that sort of situation will be dealt with. And then there is also an ethical dilemma in terms of the communication between different fields that are required to build these devices. So this is, as I said, it's a very interdisciplinary field, which requires biologists, computer scientists, engineers, physicists, and each person will bring their own expertise into such projects. And how can we ensure that there is correct communication and that we are regulating for all of the possible risks that there are in developing such devices? So there's a lot of, let's say, brainstorming and developing of plans to implement regulations, but I would say we're not there yet as to, you know, be implementing these in a commercial scale.
Yes, there is a multiple of discipline, a multiple of fields, and a mix of expertise in terms of biology, in terms of IT and other stuff. So, okay, it's clear. Okay, so thank you for the answer.
Yeah, no problem. So I wanted to also ask you a question, Lucas. So I was wondering, as you mentioned, aging and regenerative medicine and how that can be incorporated with microfluidics. If we develop all the potential of microfluidic in the aging field, do you think that we will be able to kill the biological death in the future?
Such a prospective question. And so, in my opinion, yes. I think we could kill one day the death caused by the biological processes. Microfluidic is an amazing science to understand the biological processes at the cellular level. And for me, it's a good way to more understand the biological aging, the functional aging also, and globally the longevity. However, you have a lot of, as you said, regulation. You have a work in terms of thinking, a lot of ethics questions. And in my opinion, microfluidic will accelerate the anti-aging researches for sure. However, you have a sort of a green light to develop and to adopt this kind of anti-aging solution to increase longevity and maybe a day be immortal. We don't know, but yes, it must be accepted by the population, by the politics, and allowed at the end by the regulation. So I'm pretty optimistic for yes, for the future of a microfluidic in terms of science, but also in terms of yes, in terms of societal allowing or acceptance.
Thank you for that, Lucas. No, I completely agree. I think that microfluidics can definitely contribute a lot to the advancement of all of these fields. And it would just remain to be seen at what rate or what speed it is done, because it all has to be done in a responsible and ethical way. And that, I think, may slow down these processes.
Yes, to be continued.
Yes.
Thank you. So thank you, Roberta. Thank you again for your presentation, your brilliant presentation. Thank you to the AITF Alumni Network, Krishna, and all the board working for us. Thank you for all the participants, and we will see for another meeting, I hope, in the next few months. So thank you, Roberta. Thank you all, and have a nice weekend. Thank you.
Bye.
Thank you. Bye.